Hello everyone, and welcome to today's live broadcast, Strategies for IND Filing Success. This is Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Pharmaceutical Technology and sponsored by Regis Technologies, Inc. Regis Technologies, a Chicago-based CMO focused on small molecule APIs, partners with pharmaceutical companies to expedite drugs to the clinic and ultimately to the market. They offer synthesis, analytical, and separation services to advance APIs or highly potent APIs from initial process development to final validation and commercial manufacturing. You can let their veteran project managers successfully guide your small molecule through the IND and NDA processes. To learn more, visit registech.com for more details. And we've got a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Should you have any technical problems viewing or hearing this presentation, click on the question mark help widget in the dock at the bottom of your presentation window. Now I'd like to introduce today's speakers. We're joined today by Carol Danielson, Sharon Ade, and Dan Weissmuller. Dr. Carol H. Danielson manages and serves as president of her own regulatory, clinical, and quality consulting firm, Regulatory Advantage International, LLC, based in Tucson, Arizona. She's provided regulatory expertise and leadership for more than 30 years for drugs, biologics, and medical devices from discovery through post-marketing. Her areas of specialization include regulatory strategy and submissions, clinical affairs, and compliance and quality assurance and control. Her background includes both extensive hands-on experience and corporate-level strategy activities, from partnering, due diligence, to serving as an expert witness in the drug development process. She's been pivotal in the marketing approval of numerous pharma products, has submitted and maintained numerous INDs in both CDR and CBER, and has served as lead for more than 120 FDA meetings and as a U.S. agent for several foreign firms in Asia and the European Union. Dr. Sharon Ade brings more than 30 years of pharmaceutical and biotechnology experience in specialty, branded, legacy, and generic drug pipeline delivery for global markets. She draws upon her extensive background in API procurement, analytical formulation development, tech transfer, quality compliance, regulatory affairs, and clinical development. With roles spanning CEO to Chief Scientific Officer, Sharon's provided leadership to life science companies in both technical and general management areas. Her work experience spans small startup spin-outs to mid-size and multinational Fortune 100, including industry leaders Fresenius Kabi USA, Pinnacle Biologics, Aspira, Apotek, Clinitech Nutrition, and Baxter. She holds a doctorate in pharmaceutics from the University of Illinois at Chicago, and an MBA from Lewis University, and a bachelor's degree in science from Northeastern Illinois University. And you can reach her at sade@rcainc.com. Dan Weissmuller has worked in the pharmaceutical industry for more than 10 years. Excuse me, sorry Dan, didn't put you out that long. He earned his bachelor's degree in chemistry from Benedictine University. He's currently pursuing his master's degree in quality and regulatory science from Northwestern University. He's worked at Regis since 2001. After starting in the quality control department, he's quickly advanced through analytical development, validation, and was promoted to his current position as Director of Quality Assurance. In the laboratory, Dan specialized in advanced analytical techniques such as HPLC, GC, viscosity, and mass spectrometry. During Dan's tenure, notable accomplishments include internal transfer of Regis Analytical Laboratories drug master file regulatory approvals in over 10 countries on 6 different continents, and also leading Regis through FDA pre-approval inspections without receiving any objectionable findings. So thank you to the panel for joining us today. Before we start the presentations, we want to get the audience involved with a brief polling question. So please click your answer and click Submit directly on the screen. So what is your experience in filing an IND? None, limited, considerable, with multiple filed. Again, the question is none, limited, or considerable with multiple INDs filed. So select your answer and click Submit. All right, now let's take a look at the responses. So it looks like almost 80% of the audience has either limited or no experience. So I'm sure today's speakers are going to give you some useful information. So at this point, I'd like to turn the presentation over to Carol Danielson. So Carol, please get us started.
Good morning, everyone. My part of the presentation covers the actual process of preparing and submitting in the FDA review. So basically, what is an IND? It's a notice of claimed investigational exemption. What it is basically, it's requesting permission to ship an unapproved product in interstate commerce. And what it also does, it serves as a mechanism for FDA to have an opportunity to review the contents of the application before allowing clinical studies in humans. The applicable regulations are covered under 21 CFR 312. So when do we need an IND? Well, whenever we're running a clinical study on humans, of course, in the United States, if it's an unapproved new molecular entity, or if it's a drug that has been approved but is being studied in some way that does not agree with or adhere to the approved labeling. For example, you may have nitroglycerin approved for angina. If you wanted to study it for Raynaud's phenomenon, you would need your IND. So what do we do? Step 1, application process. We prepare the contents of the IND according to 21 CFR 312. It's listed very clearly what's required. And then if we're filing electronically, we will submit those electronically, obviously, in the comprehensive contents and hierarchy format in Common Technical Document. You probably will have a pre-IND number assigned. You certainly will if your submission is electronic, or if you've had a pre-IND meeting with FDA, then you will already have an IND number. If you're filing in paper, FDA will send you a letter once they receive your IND, and your IND number will be on that. So as I mentioned, if we're using a Common Technical Document format, which is certainly most preferred, we will follow what's called headings and hierarchy, and that lists exactly where all the contents of the application need to be mapped to that CTD, where they need to be located, in other words. And this is just a breakdown. So if you look at the items, there's a form called FDA Form 1571. And on the second page, it lists the items that are required in the IND. Well, since we're going to be submitting electronically, what I've done here is I've mapped those items to the actual placement in the Common Technical Document format, the pyramid that we saw in the previous slide. So the color coding are all the items, for example, in orange that will be in Module 1, which is the regional module. And then the documents that will be in summaries and overview are here located in the light purple. Then the chemistry manufacturing, which is what we're really interested in, that's going to be in the quality section. We'll have the non-clinical here in sort of a yellow-greenish color. All of the clinical contents will be listed in that very light sort of lavender shade here. Now, as I mentioned, ideally you would want to submit this electronically. And FDA has submitted, there is a regulation that everything must be electronic for INDs after May 2018. If you're a sponsor investigator, your IND will be exempt from those electronic submission requirements. When you're doing electronic submissions, you submit those to FDA's electronic submission gateway, or you can also do it on physical media. On the left, you can see the actual formatting of how that IND submission works. You're the sponsor, of course. You submit that information to the gateway, and from the gateway, it goes on to the actual center that will do the review. You will get an automatic notice that your submission has been accepted. Okay, so once it gets to FDA, this is what your FDA review division will look like. And your main review sections are going to be medical chemistry, pharmacology, and then there's a project manager who will actually be your primary interface with FDA during the entire life of the IND. Now you see I have an arrow under chemistry. So the actual chemistry reviews occur in pharmaceutical sciences. If you're a small molecule, it will be through the Office of Pharmaceutical Quality. And if you are a biotech product that's being reviewed in CDER, it will be that Office of Biotechnology. Now, pharmaceutical quality and biotechnology are two sister offices within pharmaceutical sciences. So what does FDA look at when they're reviewing the CMC section? Well, they want to know that does the characterization, control, and stability of the drug product and drug substance support the proposed clinical study? So has your product been characterized? Your specifications and test methods, are they supportable? Your impurity profile, has it been adequately defined for the stage of development? And is your product stable enough to get through the clinical study? And are there any safety concerns caused by the molecule, the processing, the stability, any of the other issues associated with CMC? So the Office of Pharmaceutical Quality, as I mentioned earlier, that works with the rest of the review division and the CMC review. They look at all the quality attributes and the manufacturing. They do a number of other things also that are related to the CMC review. And they emphasize quality by design, and also everything is really risk-based. Now, this is very difficult to see, but the way they used to be called Office of New Drug Quality Assessments, but the Office of Pharmaceutical Quality, as you can see if you can get a blow-up of this schematic, the different divisions are represented within the schematic. So these will be chemists who will be lent to be part of the review team to the individual review divisions. And you can just take a look at that, but they are divided by actual FDA office. Biotechnology, as I mentioned, that's the sister office, and they do all the review of the biologics that are looked at in CDER. So these would be your well-characterized biologics, products like monoclonal antibodies, therapeutic proteins, these kinds of products. Now, it's important to emphasize that FDA expects to see a stepwise approach for CMC. So the agency realizes that when you start the phase 1 study, you're going to have limited CMC material or information. So they expect you, as you go through the phases, to be able to tighten that up. So you may have a very broad acceptable range early on, say for assay, and that needs to be tightened up as you get to commercialization. FDA also looks at some other sections. So they look at the preclinical, and basically what they want to know here is, do you have the animal studies to support the clinical study that you're proposing? So do you have the most appropriate species? Were the studies under GLP? Did you have enough repeat dose to support the length of the study? And were there any safety signals? Just to note that there are special concerns for biologics, and you can see those by reviewing the FDA guidances. FDA also looks at clinical. They want to make sure the investigators are qualified, that the risk is acceptable for the patients, that there's a good investigator brochure, and any other information that could pose a possible risk to the patients who will be receiving the product. Once your IND is submitted and it's thoroughly reviewed, if it's not clinical hold, you can begin your clinical study 30 days after you've been notified that FDA has filed the actual IND. So if it's paper, it will be a few days from the time you've submitted to the time FDA files. If it's electronic, it will probably be immediate. If FDA puts the IND on hold for any kind of a concern, anything they're not comfortable with CMC or preclinical or clinical, you may not proceed until the IND hold is removed. Just very briefly, once you've submitted the IND, it's a living, breathing document. You'll be providing several amendments as you go on, information amendments on CMC, changes to your protocol, and other reports. And you can see here on FDA's Form 1571, I put the arrow to where you would check the appropriate submission. Just to kind of sum up on this, the life of the IND, unlike the European Union where you'll have a CTA, for the IND, once you submit it, it continues to live indefinitely. You'll be using those contents to support your NDA, to do your post-approval studies, and perhaps even to do additional submissions as supplemental NDAs. You do have the option. You can always inactivate, withdraw, or terminate your IND. Other than that, the IND continues to be alive and well. These are just some useful guidances. If you look at the first one I have here, it's a 1995 guidance, but it's still basically very current as to what's expected in your application. So please do take a look at that. The second guidance is important for CMC. If you are opening your IND at phase 2 or 3, or once you get to phase 2 and 3, additional chemistry information is going to be required. We talked about the increasing CMC as you go through development, so this guidance is very useful for that. There are some guidances here. For example, when do you not need an IND? Some information about the regulatory requirements for the electronic submission, and also the guidance for your clinical holds if you are indeed put on clinical hold. And I think at this point, I'm going to turn it back over to Rita. Thank you.
Thank you very much, Carol. We're going to, before we move on to our second presentation, we do have another poll. So audience members, please click your answer on the screen and click submit. In simple question, which are you interested in, small molecules or biologics? Again, in which are you interested, small molecules or biologics? So select your answer and click submit. And let's see, we've got a little more than 70%, about almost 3/4 of the audience is leaning toward the small molecules. With that, we're going to move on to our second presenter. Sharon Aid is going to talk a little bit more about filing requirements. Sharon, turning it over to you.
Thank you, Rita, and good morning, everybody. My presentation will build very nicely on Carol's. The title of my presentation, the focus of it will be on the chemistry, manufacturing, and control section of the IND so that you can have successful filing at each step. My presentation outline will start with a little bit of background on the regulation of clinical trials in the United States by the FDA. Then we'll look at a very high level at what the main contents of the CMC must include for drug substance and drug product. Then we'll go into more specific detail on the CMC section as it evolves and keeps pace with clinical development. And then I'll do a brief summary. So as Carol mentioned, the clinical trials are regulated by FDA, specifically by Title 21 CFR 312.22 and 312.23, which require that the initial entry into human clinical studies in the United States of an investigational drug must be regulated under the submission and approval of an IND by the agency. There are a number of relevant guidances, and in particular, when before it was withdrawn on April 14th of 2015, the main guidance regulating INDs was titled Submission Documentation for the Manufacturing and Controls for Drug Products. But much of that was obsolete, and so it was withdrawn and replaced with a new guidance titled Chemistry, Manufacturing, and Controls Quality CMC. There is reference here to a number of guidance documents that are relevant, and I'd just like to reiterate that the two that form the basis of my presentation are the same two that Carol just made reference to, the contents and format for INDs for Phase 1 studies of drugs including well-characterized therapeutics and biologically derived products, and INDs for Phase 2 and Phase 3 studies, chemistry, manufacturing, and control information. Before I proceed too much, I want to just make a few high-level disclaimers. This presentation is applicable to small chemical entities, but it excludes novel therapeutic chemical entities, biologically derived therapeutic agents, botanical therapeutic agents, and therapeutic agents derived from natural sources. So what is included in the CMC contents? Well, basically, it's the drug substance details and manufacture or synthesis, and the same thing, but slightly different for the drug product. So if we focus in on drug substance information needed, at a high level, what the agency is looking for is a description of the drug substance, including its physical chemical characteristics. Also, the name and address of the manufacturer, the general method of preparation for the drug substance, acceptable limits, and the analytical methods used to assure the identity, strength, quality, and purity, and information to support the stability of the drug substance. Most likely this was taken during the preclinical animal toxicology studies, and to assure that it will keep the drug substance stable through the duration of the proposed clinical trial. As it pertains to the drug product, similar but more information is needed. A list of the components, including alternatives for inactive compounds used in the manufacture of the drug product, including those intended to appear in the drug product and those which may not, but if they're used in the manufacturing process at all, must be listed. A quantitative composition of the new drug product, including any reasonable variations. And at this point, there may only be one clinical trial batch that has been used, so little information may be available. The name and address of the drug product manufacturer is needed, a general description of the manufacturing and packaging procedure appropriate for the product at this point in the clinical trials, the limits and analytical methods, and information on the stability of the drug substance in the drug product, again, taken from the toxicology studies, shown to be adequate for the duration of the clinical trial, a general description and manufacturer detail of any placebo, if there is one, and a copy of all labeling. So now, with all that being said, let's talk about how this information evolves as you progress from Phase 1 to Phase 2 to Phase 3 clinical trials. The next two slides, pardon me, I got ahead of myself. The emphasis for the development and the progression during the 3 stages of clinical trials is emphasis on patient safety. The amount of information initially submitted at Phase 1 depends on the stage of investigation, the amount of proposed testing in humans, and the duration of the testing, and whether this information is safety related. So the guidances identify what information should be presented and information amendments going forward as you progress through the different stages. So at Phase 1, the sponsor must identify if the chemistry of the drug substance or the drug product or the manufacturing of it poses any risk to human patients. If it does, this needs to be discussed, and steps to monitor this have to be included. When you get to Phase 2, the sponsor is expected to document its product development program and to take this information and use it as the basis of additional controls to be able to protect the safety of the patient. So information and correlations between data generated in the IND studies and the soon-to-be marketed product have to support process controls and justify specifications. By the time you get to phase 3, the FDA encourages the sponsors to meet with the CMC review team to make sure that the initiation and execution of phase 3 clinical trials will result in information that will hopefully result in approvability of an NDA. Now, the next 2 slides contain quite a bit of information. I do not intend to go into detail on this. It is presented so that the reader will have an access to an idea of scale of how this information is intended by the agency to evolve with the knowledge that is taken from the clinical trials. So if we just look at a couple of representative rows here, I'll highlight some of the differences. In row 1, we're talking about the physical and chemical characterization of the drug substance. So when you go into phase 1, a brief description and some evidence to support the chemical structure is probably sufficient. However, by the time you're in phase 2, details on the chemical structure must be available to help predict the possible metabolites, and if this product is a peptide, the amino acid sequence and peptide map essentially would need to be available. By phase 3, very specific details, including dissociation constants, isoelectric points, hygroscopicity, crystallinity, or evidence of amorphous structure, etc., are needed. So you can see how this information increases over time. If we step down to row 4, applicable limits and analytical methods, at phase 1, a brief description of the test methods and acceptable limits is probably adequate to be able to start the study. But when you're in phase 2, like Carol was saying, critical quality limits have to be defined for the stability batches, and by now there should be lots of clinical trial material, different size batches, and different degrees of stability information that would be available to support the limits and the analytical methods. And by the time you're at phase 3, a very detailed list of all of the methods would be necessary. So if we move forward to drug product, we can see the same thing happening. If we look at row 1 on the composition and the components and inactive excipients used in the manufacture, a brief introduction of maybe 1 to 2 pages is necessary at phase 1. By the time you're at phase 2, all components used in the manufacture of the drug product must be identified with specific reference to a quality reference, such as the NF or USP. And at phase 3, full characterization of all of the information related to a DMF or a drug master file are expected. If we jump down several rows to applicable limits and analytical methods, by now we are looking at much more information again. So at the beginning in phase 1, a brief description of the analytical methods, including some limits, is acceptable, but by the time that you're in phase 2, physical-chemical parameters have to be identified, impurities have to be understood, viscosity, other attributes based on the evaluation of the formulation and its applicability to patient safety have to be identified. In phase 3, all information is required to be known and disclosed. Specific degradation pathways of the drug substance and the drug product in its particular container closure system for the duration of the clinical trial would be used to justify this. In summary, what I'd like to say is that the content of the CMC section of the IND evolves over time, and it evolves in parallel with the product development process. This happens in order to better meet the clinical needs of the patient. The amount of detail required in the CMC section needs to be appropriate to address relevant safety concerns to the patient for the duration of the clinical study, and the FDA encourages sponsors to interact with the agency at each clinical development stage so that the quality of the data progresses in line with what will be needed to obtain a successfully approved NDA. And that will conclude my presentation, and I would like to turn it back over to the moderator.
Well, thank you very much, Sharon. Before we move on to Dan's presentation, we do want to have one final poll question. So once again, please select your answer on the screen and click Submit. And we need to back up one slide there. All right, thank you. So what dosage form do you plan on using for clinical trials? So inhalation, oral tablet or solution, intravenous, or topical? So again, the options are for the dosage form you plan on using for clinical trials: inhalation, oral tablet or solution, intravenous, or topical. So please make your selection. Click Submit. And now we'll take a look at the answers. And there we go. And oral dosage forms seem to be for about half our audience. But we do have some representation for the other categories also. So with that, I'd like to turn it over to Dan Weissmuller, who's going to talk about analytical methods and validation processes that are needed for successful filing. So Dan, please start your presentation.
Thank you. Welcome, everybody. So my goal here is I'm going to try to take some of the concepts that have been brought forth by the previous speakers and try to bring them into the manufacturing. In particular, I'm going to focus a little bit on the drug substance and API. As you recall from my experience, I've been here at Regis for actually 15 years now with an analytical background, and I've collaborated on about probably 50 INDs, the scale-up and development of new entities as they're coming through the plant here, and help support those. And some of the success stories is taking at least 3 of those through NDA into about 50 different countries. So I'm going to try to take some of that experience and then crystallize that down into some API case studies. So we're going to start by an introduction, just a brief review of some of the pharmaceutical development concepts. We're going to talk about some of the planning that's going to need for success pre-IND. And we're going to talk about the goals, which is process validation, and then commercialization, getting ready for your NDA. We're going to go through those case studies and with a focus on the analytical, the specifications and validation that's required along the way at each of the major milestones. And then, if time permitting, we're going to get into a few other concerns, things like cleaning validation, equipment qualification, PGIs, and maybe some other hot-button pitfalls, common pitfalls that people see with their CMC and the analytical. So once again, another look at the pipeline. I'm sure many of you have seen all different representations of this, but I'm using basically the same baselines that the speakers before me. I mean, these are great resources, the guidelines for writing INDs, and information for the manufacturing and the analytical is throughout these. But in addition, when it comes to the analytical and the manufacturing, the concept of GMPs also applies. So one of the predicate rules here is the Code of Federal Regulations, Title 21, Part 210-211. That's the good manufacturing practices for pharmaceutical products. And that pertains to the focus here, where we're talking about the small molecule, the chemical synthesis pharmaceutical products. Along the way, the problem with that, though, is that those laws and regulations are written with commercialized product in mind. So the FDA has provided these guidelines to help fill the gaps and help us better understanding how to phase in all that information and what's necessary along the way. So there's another guidance for industry, CNGUP for Phase 1 investigational drugs, that was written a little more than a decade ago, and I've got references at the end. And then ICH-Q7 and a variety of the other ICH-Q series of guidelines and guidances are fantastic for helping develop specifications, develop analytical validation, and help understand impurities and what needs to be done about them. One important note is that when you do look at these ICH-Q guidelines, is to be very careful. Like, for instance, with Q7, It's about a 50-page document, and only the last 2 pages really pertain towards clinical API, whereas a lot of it is written towards commercial product. ICHQ-11 is another great document to look for the development of pharmaceutical products and drug substances and what needs to be done. So, moving along. So planning for success starts pre-IND. So the IND requirements is that we're going to have a brief description of the manufacturing process, including a list of reagents, solvents, and catalysts used. It's going to have flow diagrams, and you're going to list out the methods and the specifications as well. So one of the goals here is that as you're preparing for your IND is that you're going to have to start developing the chemistry, and there's going to be sufficient development to remove harmful impurities, right? Because now we have a consideration for patient safety, where initially we're trying to manufacture these products, evaluate them, get a sense of what might be a potential good candidate to take into a clinical trial. Now we need to make sure that the route that we're using for manufacture doesn't have any issues with it that would interfere with or potentially be toxic themselves. Examples of that might be harmful raw materials, intermediates, or byproducts. So you're going to want to list those out and get whatever information you can to start understanding those. That might be using modeling software to get a sense if things are potentially genotoxic or mutagenic impurities, and you might want to take a look at any sort of data that might already be out if it's a commodity chemical and such. There's also going to be a quantum change that in scale of manufacturing at this stage, where previously a few grams of material might be all that was needed. To get ready for a GLP study, you have to consider that it might be a few hundred grams to do the study itself, but then also for all the analytical and other requirements that you might need 30 grams for, say, release testing. You might need multiple sets of that 30 grams to do any sort of stability testing. Then there's going to be retains and reference samples you might want to be collecting along the way. So it means that you're moving from the gram scale to the small kilogram scale. And with that becomes also a requirement for process safety in addition to patient safety. So just be mindful that that's something that's going to have to be considered. It also changes the availability of starting materials, raw materials, and some of the goods that you're going to want to be looking for. So process validation really is the goal from the analytical perspective. It's getting the eyes and ears onto the process and getting things to the point where being mindful of the endpoint. So there's going to be a number of reports and outputs that are going to support the CMC sections of the IND. These process development reports, completed batch records of summarizing the details of manufacturing campaigns, engineering reports, and then for the focus moving forward here for my presentation, it could be these analytical development and validation reports, certificates of analysis, and stability summaries. And all this is going to be important to show that the drug that's being made is being made consistently. It's being made in a way that's going to meet the benchmark for safety and provide product that's meeting its label. So as we move forward, we're going to talk about the tox batch. So where this all starts is with the preclinical. Before we get into the case studies, I just want to stop here and talk about this for a minute. Tox batch, GLP batch, these are really slangs used to refer to the material that's going to be used in the IND-enabling animal studies. The goal for this material is to be well characterized, well understood. So there should be a deal of work to make sure that you have the material that it's intended to be through NMR, infrared spectroscopy. You're going to want to know its purity, so some sort of chromatography, whether with its UV detection or some orthogonal mode that you're going to understand how pure and what quantities certain related substances are present. Because ultimately this is going to set the baseline for quality moving forward. So these animal studies are really the first data that you have in place to benchmark the materials that you're going to be bringing in for people in their clinical trials. This also should be well documented, so just keep in mind that being the benchmark, you're going to want to be able to find that information and get back to it. So while this isn't really a GMP requirement at this stage, make sure that you've got this under well control, that it's being well documented, that you're getting the reports in a way that you can refer to that information easily. So moving into specifications, as you're setting the specifications, that's really going to be determined by the route of administration. So we talked about understanding the material. General test, appearance, identification, related substances. Related substances could be relating to intermediates, raw materials, starting materials, byproducts. Organic volatile impurities, such as solvents used in processing, are important to understand and review. Inorganic impurities, these could be catalysts, residual metals. They could be certain heavy metals that could be present in your starting materials, like the lead, mercury, cadmium, arsenic concerns from ICHQ3. If you have a salt formation, you're going to want to understand perhaps your counterion to establish your stoichiometry. And if there are any chiral centers in your molecule, this is also an important thing to understand. For oral dosage, some form data should be being collected and understood. This might be critical if there's polymorphs or solvates or different types of forms that can be formed for your product. For topical injectable parenterals or ophthalmics, you're going to understand bioburden or endotoxin. And again, for the ophthalmics, clarity is an important test for certain markets, such as the EU. For raw material, working backwards in the process, the requirements are a little more relaxed, starting with drug product, of course, being the most strict, and then working back to the drug substance, and then before that. So for raw materials and intermediates and key starting materials are typically minimally examined for their purity and ID, and then the raw materials, it may be more appropriate to just work with a C of A, but with a minimum doing ID testing. But keeping in mind that when you're going through pharmaceutical development, especially at early stages, specifications set by vendors and by third parties might not be appropriate for your process. So be mindful. It's a common pitfall. Use tests may be required or be in your best interest. And use tests or front runs would be actually taking the materials you have and testing them forward through your process to make sure that there aren't going to be any issues downstream. So one specification isn't necessarily, for somebody else, isn't necessarily good enough for you. So carrying forward the themes that we discussed before, validation requirements increase over that pharmaceutical product life cycle. So going from preclinical into phase 1 and then phase 2 and then phase 3, the requirements increase. And the same is true as you go through the pharmaceutical process, the starting materials, the intermediates, and then drug substance and finished product. The requirements for validation increase, and they're brought in more quickly at different stages. So this table is designed to sort of highlight that. And while it might be a little difficult to read here, just sort of the general themes being is that with finished product, preclinical, you're going to want to have a well-developed specific assay, something that's, we're moving off of the HPLC system where we ran a dozen compounds on a C18 column that you've trusted for 20 years. And we're working, it's best to have a method that you've demonstrated some of the analytical performance characteristics, such as accuracy, precision, specificity, linear range, sensitivity. And while it might not be necessary to validate these methods at this stage, you certainly want to understand that these methods are validation ready. As you get into clinical, phase 1, phase 2, that validation for the finished product in drug substance should be implemented. And in phase 3, you'll want to have you'll have completed validation on your starting materials, intermediates, and finished products. And as we move forward here, we're going to talk a little bit about the fact that validation requirement changes as well. So moving into our first case study, and then we're going to kind of move through these quickly. The quality target here is a well-characterized materials we talked about, avoiding known risks. Product-specific release assays and related substances. Organic and inorganic impurities should be understood with ICH limits in mind, and that's the ICH quality guidances that we talked about. Q3 is ICH Q3. If you haven't read it, that's a great place to start. Validation requirements are typically at this stage informal. Development summaries are appropriate. Typically, the parent compound is used as a surrogate for any sort of validation readiness assessments. Limited impurity data might be available. Your route of manufacture really isn't locked in at this stage. There could be some change there. But you're going to want to evaluate any intermediates or raw materials that you're using, things that are like difficult to detect or separate. So you're looking at clinical pairs and you're understanding that your method is specific so that you don't run into new impurities at a later stage that you just missed in your analytical. So if we move forward, so here's an example of a sort of a table, what a C of A might look like for a GLP batch, where you'll be doing appearance, some identification testing. This is a case study for, say, a small molecule API that, or active pharmaceutical ingredient, drug substance, that would be used in an oral dose product. So you can see there's identification testing. We do an assay by HPLC. One of the themes here, though, is to look at specifications aren't firmly set at this stage. They're typically reports. We're looking to sort of gather this information with some targets in mind for things that we think the FDA may have less questions if you have a well-isolated compound, like 98%. If you start working with lower purities than that, it's just going to put the more burden on you to understand what the other components are. But ultimately, to set the baseline, you need to know what the information is. So at phase 1, phase 2, specifications start to get set based on the toxicology data. And one of the quantum changes here, though, is that we're going to start needing to have stability data. So stability methods must be proven stability indicating, where we stress test our materials and then show that what they're going to fall into, what's going. This is actually going to be sort of the baseline data then later on for supporting whatever stability commitments are going to be made in the IND. For validation, it would be required for the finished product, but not necessarily for starting materials and intermediates at this stage. API drug substance also as well should be considered for validation and for the release tests. But however, this validation at this stage is a little bit more limited. The parent is typically still the surrogate, and intermediate precision, such as testing multiple laboratories, multiple analysts, may not be appropriate. So looking at that same case study, now we've started entering in specifications, and these specifications would have come directly from the tox batch and whatever sort of GLP limits and commitments have been made to the FDA. And you can see here that since this would be specifications for a drug substance, we've started validating our release assays. So for phase 3, this is actually pretty simple at this stage because it's everything at this point should be being evaluated and validated. So that's all of the analytical performance characteristics, the accuracy, precision, linearity, range, sensitivity, specificity. And at this stage, also intermediate precision and some robustness data should be generated along the way. The validation requirements at this stage, so whatever that may be from your laboratory, might be a 2-analyst, 2-day, 2-instrument, 2-facility, 2 plants, so that you're understanding that this method could be transferred. Impurities are also well understood. Reference standards in place for these as well at this stage, so that you're understanding your relative response factors, that you're not being fooled into misreporting or misquantifying these types of risks. So looking at this final case study, then you can see that the specifications then again are still validated. And I've added to this section on related substances that includes the impurities, that those are now included in there because your process is locked and there shouldn't be any changes at this stage. So last, for some of the other points of interest, analytical equipment. Really, the qualification of this should be in place at the point where you get to the toxicology batch. While it might not be a listed requirement in many of the documents, you need to rely on this data, and it is the input into your IND as far as safety. So it is highly recommended, and then later on as you get into your clinical studies, phase 1 and beyond, that it is a requirement that it's qualification, IQ, OQ, PQ, those terms are all completed. So for cleaning validation, that's another analytical consideration. If you're in a multi-purpose facility, that's going to start right away because it's going to be required for product changeover, but in a dedicated facility, it's typically done during PQ. That's making sure that you have the tools and your analytical tools are tested to show that that residues are removed from your manufacturing trains. And then just again to touch on PGIs, this is a really important topic these days. There's a number of the FDA will be reviewing the intermediate starting materials, raw materials in your processing throughout phase 1, phase 2, and assessing these using modeling software. So you need to be doing that as well to avoid holds for potential risks in your products. And this analytical can be difficult, so you should have a plan for this going in. Very sensitive techniques such as mass spectrometry might be required, and these methods take time to develop. As you get into late stage, this also can be considered for solvents, and another common pitfall I've been seeing on more reviews lately is understanding PGIs in your raw materials is potential to be in your final product. So that might be, say, if you're using large volumes of chlorinated solvents, that you might have Class 1 solvents present in those and being able to justify how they're removed. So there's a page for the references acknowledgments, and just to say thank you for your time, and I'll turn it back over to you, Rita.
All right. Well, thank you very much, Dan, and thank you to all our speakers. We'll now move on to our Q&A session, and just a reminder to the audience, you can submit your questions by typing them in the Q&A box on the right-hand side of your presentation window, and we'll try to get to as many questions as we can. So our first one is for Carol. Is Module 2.3 required for IND submission in addition to Module 3, or is Module 3 enough for IND?
That's a very, very good question. I typically include in Module 2 just to give an overview, but basically if you look at the mapping, it appears that you can present everything in Module 3. However, I would strongly recommend Module 2. It's very, very good for the reviewers. Of course, you do have to have a general overview of your application, which would be in Module 2. As far as the 2.3 section for the quality, I would definitely include it. It's so much easier for the reviewers, and I've typically seen that done.
All right, thank you. Sharon, here's a question for you. Do all clinical drug product lots have to undergo stability studies strictly according to the protocol filed in the original IND? Sharon, are you there?
The answer is yes, as long as those batches are being used in the clinical trial as the actual drug product being given to the patient. In the early stages, there probably are only enough clinical drug product lots manufactured as are needed. So I would say that generally the answer is yes.
Okay, great, thank you. Dan, with regard to tox studies, are you saying that the material manufactured for tox studies should be manufactured under GMP and the tox studies should be conducted under GLPs?
So, good question. I'm sorry if I misled a little bit, but so it's not required to be manufactured GMP for the batch that's going to be used in the animal studies. But I would recommend that this be well documented because you're going to want to understand how it was made so that you can reproduce that benchmark. As you move into phase 1, there is going to be a GMP requirement. And in some markets and for some companies, there are strategies to use same material for GLP in phase 1. If that's going to be the route you're planning to take, you should consider making that material GMP because you're going to have to go to the lowest common denominator. So if you're going for a phase 1 batch, that must be GMP. So you'll have to manufacture the tox batch or the GLP or tox batch GMP in that case. But if you do that route, be mindful that that pre-work, that process development to work out to make the manufacturing route pharmaceutically safe, getting out some of those toxic solvents and reagents, is going to have to be done a little bit earlier because that's required to get to that standard.
Okay, thank you. I'm going to start with Carol on this question, but invite the other speakers to also comment on this. Does the EU have different or additional filing requirements? Carol, you want to start there?
Sure, that's interesting because we're actually talking about the IND versus the CTA at this point if we're talking about the very early stage. So the information that goes into the EU, it's the investigational medicinal dossier and the actual the CTA section, the clinical. If you map what's required for the IND against what's required for the CPA and the investigational medicinal product dossier, most of the contents are the same. There are a few differences. There's a risk section requirement in the EU, but generally you could typically take most of the material and go back and forth between the two pretty easily. They're just placed in different sections with a little bit of a different emphasis. For the CMC, I'd like to turn it over to my CMC experts, the other panel members.
Yeah, Sharon, do you have some comments on that? Any EU differences?
There are differences, but I would not say that I am an expert in that field, so I would defer back to Carol on that.
Okay, Dan, have you experienced, can you talk a little bit about your experiences working with EU?
So I can say from a manufacturing perspective that there are a lot of similarities. Generally, where I've seen a lot of more differences is on the tail end as far as the review. But ultimately, so that's kind of a broad conversation. There are some differences from the CMC side, but again, there's less involvement or direct involvement, I would say, from my perspective, being a third party. So I would kind of deflect that question because maybe I can get a little bit more specific.
Mm-hmm. All right. Well, thank you. I have another question for you, though. How much stability is required to open an IND?
Who are you addressing that to, Rita?
That's for Dan.
Okay.
So for opening the IND, you're going to have a stability protocol, obviously, for any of the materials you're going to put on the clinic because that's going to be required. And then leading up to that, you should hopefully have stress test your molecules to understand what the potential risks are and to justify and support your plan for what would be appropriate storage and handling of that compound to that point. So it's going to be less data than later on. You're really kind of collecting stability data as you go. And it's going to matter for how long you intend to use this. So if you're going into a toxicology study, obviously you don't need long-term stability for that, but the material that you're going to put into the clinic is going to have that. So that protocol and plan and the justification for why that plan is going to be needed is important. And if Carol and if you or Sharon have anything to add there, I'd invite you to do so.
Yes,
I'd love to add something. Oh, go ahead, Sharon.
I was going to say that I have had first-hand experience starting out some simple I shouldn't say simple, but some basic bioequivalency studies or PK/PD studies. And these are considered phase 1 human clinical trials. We had to have enough stability data to be able to complete that study. And so if the study lasted 3 months or 6 months, then generally you need to have a minimum of 3 to 6 months stability data to cover that. This increases as you go through into longer pivotal studies, in which case you might have to have a minimum of 1 year. And if the study runs more than 1 year, then you might end up in the unfortunate situation of having to manufacture a second batch of clinical trial material to extend past the year unless your retest date allows you to extend the 12-month expiration on the first batch. Hopefully that makes sense.
Okay, I need to add something to that also, if I may. It's Carol. The amount of stability will depend, as Sharon just pointed out and so did Dan, on the length of the study. But basically, with a small molecule, if you have a lot running ahead of the lot that you're actually using for the clinical study, you can get into that clinical study with a minimum amount of stability because you're being supported by that previous lot, even if it's a non-GMP lot. It's expected, of course, that you'll have enough stability to show that the product doesn't fall off as you're gathering additional stability. So I've seen situations where you can get into a 3-month study with 6 weeks of stability on the assumption that you will have the data to support that as you get to that point in the study.
All right. Well, thank you. Unfortunately, we are out of time. I thank all the speakers for chiming in on these questions and adding to the discussion. I want to thank them, speakers, for their presentations and the audience for their questions and involvement today. I'd also like to thank our sponsor, Regis Technologies, for making today's webcast possible. This program will be available for on-demand viewing through October of 2017. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. We invite you to forward that announcement to any colleagues who may have missed today's live event. Again, thank you for joining us, and we'll see you next time. Thank you.